very pleased to welcome Dr. Heidi Jacobs to the UCSD Shiley-Marcos ADRC Neurodegeneration Seminar Series. Dr. Jacobs received her doctorate in clinical neurosciences from Maastricht University in 2011 and is currently an associate professor of radiology at the Harvard Medical School. I picture at her early to early mid-career stage with more than 130 publications, 17 active grants by my count, and totaling millions of dollars in current funding. And she has an astounding H index of 45, which is a reflection of the high impact of her work in the field. She's received a number of honors and awards for her work, including recently the prestigious De Leon Prize in Neuroimaging in 2022, awarded by the Alzheimer's Association for selection as one of the best neuroimaging studies of the year. She's the current chair of the Neuromodulatory Subcortical Systems Professional Interest Area of ISTART for the Alzheimer's Association, and is highly sought out as a collaborator with no less than 9 pending grant applications, which attests to her high affinity for other researchers wanting to scoop her up and include them or include her in their work. We're pleased to have her join us this morning to talk about some of the work that she's been doing for the better part of a decade, I think, on the role of the small brainstem structure, affectionately called the blue spot in Latin, or locus coeruleus, and its role in Alzheimer's disease pathogenesis and progression. The title of her talk this morning is Locus Coeruleus Neuroimaging, the Road to Detect the Pre-preclinical Stages of Alzheimer's Disease. And please join me in welcoming Dr. Heidi Jacobs.
Thank you so much, Mark. That's a very flattering introduction. And thank you so much to also Dr. PD and Dr. Gigliotti for inviting me here today. It's my pleasure to share with you some of the work that we have been doing and some of my fascination with the locus coeruleus. I will share my slides again with you. Here we are. I am on a Mac, so I don't always see all the questions that may come in on Zoom. So if there's something that I'm missing, feel free to interrupt me. But I do believe we have also time at the end to answer some questions. So yeah, I wanted to talk with you about the locus coeruleus, my fascination for the last decade or so, and how we can use that information to detect the earliest stages of Alzheimer's disease. I have no real relevant disclosures other than that I am indeed the chair of one of the professional interest areas of ISTART, and I'm also an advisory board member there. But this is not relevant to the work that I will be presenting to you. And since I am presenting for the ADRC, I think this is common knowledge to everybody here that Alzheimer's disease is neuropathologically described by the accumulation of the beta amyloid protein as forming plaques, and then the accumulation of the tau protein forming tangles. And these two neuropathologic events have led to the research criteria of Alzheimer's disease that we are currently familiar with. I know that there's an update in progress, but overall, the way we define Alzheimer's disease in research is based on the presence of these two neuropathologic hallmarks, either measured in cerebrospinal fluid, in using PET imaging, and nowadays also with plasma, and combine that with with problems in daily life functioning and cognitive dysfunctions. This is how we define Alzheimer's disease with dementia. Now, in our attempt to identify Alzheimer's disease as early as possible, we've been trying to figure out ways that we can describe earlier stages of Alzheimer's disease. Because we know that these plaques and these tangles, they accumulate at least 10 to 20 years before we see the first clinical symptoms. And so in the 90s, we came up with the concept of mild cognitive impairment that has now been termed prodromal Alzheimer's disease, when individuals have presence of one of these neuropathologic hallmarks, and when they have mild symptoms in cognitive function. And since 2000s, we have now progressed to even earlier stages of Alzheimer's disease. And we've termed this as preclinical Alzheimer's disease. So those are the individuals that are asymptomatic, they're cognitively normal, but they do have evidence of amyloid beta pathology or tau pathology in their brain. And this is where we are at the moment. Many clinical trials are trying to screen for preclinical Alzheimer's disease. The A4 trial, the AHEAD trial is trying to do this, where they're focusing on preclinical Alzheimer's disease and try to treat those individuals with the idea that if we treat earlier in the disease process, we may be more successful because we are actually interfering in a stage where these pathologies have not yet done so much irreversible damage to the brain. Now, within my group, we are trying to push this a little bit further. As Mark already mentioned, we are trying to go to what we refer to as the preclinical stages of Alzheimer's disease. And this is the group here in green. So these are cognitively unimpaired individuals, also asymptomatic, but they are not yet considered to be amyloid positive based on our current biomarkers that we have. So that's a huge group of people, of cognitively normal older individuals that can also be in midlife, where we are trying to figure out who is likely to progress to preclinical Alzheimer's disease and who is likely to be on a path of more successful aging. Now, when I started out with this work, people said that this was crazy, this was impossible, because how far can you go? How early can you go in trying to detect Alzheimer's disease so early? And I always said, well, I think we as humans are really good in predicting things. And I love to show this painting that comes from what was painted in 1937 by Umberto Romano, which is the settling of Springfield. This was this settling of Springfield occurred in the 17th century. I'm not here to talk about the history of this painting, but I would like to point out that even in 1937, people were good in predicting that nowadays people love taking selfies of themselves. So I would say, if they were able to do that in 1937, we should be able to predict Alzheimer's disease 30 years ahead. And so in doing so, I have been guided mostly by autopsy studies. I consider that as the gold standard for the work what we do. And this has led me to the locus coeruleus. And for those of you who are less familiar with the locus coeruleus, it's this nucleus. Can you see my mouse? Yes, I think so. Yes. So this is a small nucleus in the pons here. It's very tiny. It's about 2 by 2 millimeters thick, and then 15 millimeters long in the healthy brain. So it's really small. It has only 45,000 neurons in an adult, and it provides norepinephrine to the entire brain. Now, as I mentioned, the reason that I am so fascinated by this structure is because of the neuropathology literature. When you look back at the neuropathology literature, and these are the Braak stages of tau pathology, originally in the 80s, when Heiko Braak presented his tau pathology stages, he described the entorhinal cortex as the first site of tau pathology. Then in 2011, he examined over 2,000 autopsy brains across the adult lifespan, so from childhood till adulthood, till late adulthood. And he discovered that there are individuals who have already tau pathology in the locus coeruleus, here in the brainstem, without having any concomitant pathology in the cortex. And this was in individuals of age 20 to 30 years, where we were already be able to see some hyperphosphorylated tau accumulations in the locus coeruleus. And the assumption was that tau pathology from the locus coeruleus would then propagate to the entorhinal cortex by around age 50, through neuron-to-neuron neurotransmission, and from there it would then spread to the entire limbic system and the cortex. And as soon as it leaves the hippocampus, the medial temporal lobe system, this is when amyloid pathology would also come into play. So then individuals are considered to be around age 60. And when you look at amyloid pathology, it has actually quite the opposite pattern of accumulation. So for tau, we see this bottom to from brainstem subcortical to cortical progression. For amyloid, we see quite the opposite. We see that it starts to accumulate in basal forebrain areas, then the rest of the cortex, and then ultimately to the brainstem and the cerebellum. And so this is what brought me to the locus coeruleus. If this is the first site in life in adulthood where we can see tau pathology, then that may be the key for early detection. We just need to be able to visualize it. And this is where the problem occurs. When we look at current tau tracers, they can only start to detect tau pathology in the entorhinal cortex. We do not have the ability to measure tau pathology in the locus coeruleus yet. For a few reasons, this is due to off-target pathology, off-target binding, but also due to the size of the locus coeruleus. So we are in Braak stage 1 and 2, where we're talking about tau pathology measured with PET. With amyloid pathology, we are usually in the thal phases 2 and 3 when we have enough binding to be able to detect it. So that means that we need to figure out other ways to image the locus coeruleus. And what do we know about the locus coeruleus? As I mentioned, it provides norepinephrine to the entire brain. Because of that, it's involved in many cognitive functions and behaviors. Think about emotional memory, attention, arousal, but also sleep and several neuropsychiatric symptoms. It is known to play a key role in neuroinflammation, in vascular reactivity. But as I mentioned, it is really, really small. So that makes it hard to image it with standard imaging methods. It is also close to the fourth ventricle, so this area here. And the fourth ventricle has a lot of arteries that are located there. So there's a lot of pulsatility of vessels as well that cause motion, that cause physiological artifacts because of that. And if you look more closely at locus coeruleus, it becomes also more diffuse in its caudal direction, making it harder to image the entire length of it. And for those of you who have been close to an MRI scanner, when people are being laid down in the MRI scanner, usually the coil stops around here. So that means that you are close to the brainstem, where the signal to noise ratio of what you can image is becoming worse. So we need to find new methods to do that. And so one of the core research questions that have been guiding me in all my work is, can we use in vivo locus coeruleus imaging, assuming that we can do proper work of that, become a marker that signals early risk of Alzheimer's disease? And so as soon as we have a method that we can use to image the locus coeruleus, I think what we need to show in order to say that this can signal early risk of Alzheimer's disease is that it needs to correlate with initial tau in the brain. So we need to show that it follows almost kind of the Braak staging pathology, where you would see a strong correlation with the entorhinal cortex. We need to be able to show that changes in the locus coeruleus precede changes that are happening in the cortex. If we follow the Braak stages. It would be great if we can also get some insights into how tau spreads, because that can then help us to develop new interventions and figure out ways in how we can stop spreading. It needs, of course, to track with the early cognitive symptoms of Alzheimer's disease. And as I mentioned earlier, when we are looking in these preclinical stages of Alzheimer's disease, not everybody develops Alzheimer's disease ultimately. So we need to find pathways that confer resilience, that indicate who is protected from progression and how the locus coeruleus plays a role in this versus who is more at risk. Now, to start off with imaging the locus coeruleus, and I know that Mark and Jeremy are very familiar with this work. So when you're trying to image such a tiny structure in the brain, it always helps when you try to search for tissue mechanisms or biological components of the tissue that can help you generate contrast in an MRI scanner. And so here you see an axial slice of the brainstem. This is postmortem. And this is the locus coeruleus. So this is a staining with tyrosine hydroxylase. And so this is coloring all the locus coeruleus neurons right here on the left and the right side. The locus coeruleus, interestingly, also accumulates neuromelanin cells throughout life. And when we look at some of these immunohistochemistry studies, we see that the neuromelanin cells, here depicted in the dotted line, are basically absent early in life and then start to accumulate until midlife and decline again. This is because neuromelanin cells, we're not born with them, but they develop as we are involved in many things that force the locus coeruleus to be active. So when the locus coeruleus is active, it generates a lot of byproducts, including iron, copper, and those byproducts need to be sequestered, and this is what neuromelanin cells typically do. So they make sure that all of those byproducts are cleaned up. And this is why you see an accumulation of neuromelanin cells throughout life. And the assumption is that it starts to decline again because of neurodegeneration. This is in contrast to norepinephrine cells that have a more linear association with age. Now, the fact that these neuromelanin cells sequester iron and copper has led to the idea that this may actually help us to generate MRI contrast, because we know that iron has paramagnetic properties. And this is exactly what research have been trying to do. So here you can see these neuromelanin cells. You don't need any staining for them. You can just see them by postmortem tissue. And if you develop sequences that try to tackle that specific tissue contrast, you get this hyper-intense signal in the locus coeruleus. Here, this is also postmortem data. And this has been done by several groups right now that all have shown, again, this inverted U-curve of that signal intensity with age, very similar to what was shown in the immunohistochemistry studies. Now, the real biological source of this contrast is still very much under investigation and debated, but there is a consistency in seeing these inverted U-curves. And so we at MGH have also tried to image the locus coeruleus using some of those sequences. And our goal was to develop a sequence that was short so that we would mitigate some of those physiological artifacts, but also motion artifacts, and that it would be clinically usable. So this is now a sequence of 3 minutes and 20 seconds. And you can see locus coeruleus intensity here in every individual across the lifespan, including here in our MCI, cognitively impaired individual. And so what we do is we extract that signal and we use that in our subsequent analyses where we are then exploring some of those conditions that we would like to verify in order to see that locus coeruleus imaging can signal early Alzheimer's disease risk. So we have implemented this sequence in the Harvard Aging Brain Study here in Boston, which is a longitudinal cohort study. This was mid-study. So at that time, individuals were around median age of 75. There's slightly more women, as you can see. And we had 32% of individuals who, according to the research criteria, would be diagnosed as in the preclinical stages of Alzheimer's disease. We also had tau PET imaging, and 22 individuals were considered to be cognitively impaired based on CDR rating. So based on all of this information, the first step we try to do is to see if we can verify that inverted U-curve that other studies have reported. And so these are the different groups that we have included and scanned. And you can see that as individuals get older, the intensity in that locus coeruleus signal extracted from the scans decreases and is also lower in individuals who are cognitively impaired. And we indeed saw that inverted U-curve as well, just like other studies. What we did notice was that there's a huge amount of variability in that older group. And this was something that we were wondering about what it could mean. And so what we did is we decided to look at whether the presence of amyloid or tau pathology could differentiate some of these individuals. And so we had PET imaging for most of the individuals above age 50, as you can see here. And what we saw was that individuals who were classified as being either amyloid negative, here in blue, or tau negative, so here in the right scatter plot in blue. So those who are amyloid or tau negative had actually a flat association with age, but it was those individuals who we would consider to be amyloid positive or tau positive that showed this downward trend with age. So suggesting that locus coeruleus intensity can signal some burden of pathology in the cortex. Now the question is, do we also see some spatial topography to this? Does this resemble some of the Braak staging that I discussed earlier? And so when we do vertex-wise analyses, so where we correlate the signal of the locus coeruleus with every voxel of the tau PET scans, we see this negative correlation here in the entorhinal cortex and its surrounding regions, both on the left and the right side of the brain, showing that lower locus coeruleus intensity correlated with greater entorhinal tau pathology. And this was also true when we only looked at individuals who were cognitively normal, or when we corrected for amyloid burden, or when we corrected for atrophy. So that's great. That shows us that with locus coeruleus intensity measures, we can actually track some of these early stages of tau pathology that follow the Braak staging. But can we also say something about its predictability for Alzheimer's disease-related cognitive decline? And as I mentioned, we started to acquire the sequence MIDS study. So we had a lot of retrospective cognitive data of these individuals. Here you can see some of those trajectories for the amyloid negative and the amyloid positive individuals. And as many of you know, cognition is always messy. You can see here the messiness of those scores. But in the overall trend, you can see that individuals who have low amyloid burden show a slight practice effect, whereas individuals who are amyloid positive do not show this. And so using that information, combined with our locus coeruleus intensity metrics, we found that at a given level of amyloid pathology, those individuals with lower locus coeruleus intensity measures here indicated in orange showed a faster cognitive decline than those individuals who had higher levels of locus coeruleus intensity. And what was really interesting here is that we saw these effects become significant at an amyloid level that is lower than where typically the Harvard Asian Brain Study puts the cutoff for amyloid positivity. So the amyloid threshold for positivity is here in the red line. So in Harvard Asian Brain Study, it's 1.32. But in our analyses, we saw that with locus coeruleus intensity measures, we can detect amyloid-related cognitive decline earlier than that threshold, suggesting that we may be picking up of some of the earliest amyloid-related cognitive decline with locus coeruleus intensity measures. But of course, all of these analyses are cross-sectional. and they remain at the associational level. So one of the things that has been really keeping me awake at night is, well, what really comes first? Is it the locus coeruleus? Is it cortical tau pathology? And this is some of the work that we've been working on more recently. This is work by Ellie Bucheka, a postdoc in our group. And so she has been working on some of our longitudinal data. So far we have data collected. This is also Harvard Aging Brain Study data of 72 individuals who have had 2 sessions of the PET scans, so tau PET scans, as well as 2 sessions of the locus coeruleus intensity metric, separated by on average of 3 years. And she correlated changes in locus coeruleus intensity with changes in tau pathology. And we found that those correlations led us to the medial temporal lobe. So you see here these correlations in hippocampus, the amygdala, indicating that declines in locus coeruleus intensity were associated with increases in tau pathology in these medial temporal lobe regions. Now, to get some idea of the directionality of those associations, we split out the regression analysis in 2 ways. So we correlated baseline tau pathology in the medial temporal lobe with follow-up locus coeruleus intensity. So that would not be what we would expect based on the Braak staging. And those correlations you see here in gray. And then we compared that to correlations where locus coeruleus at baseline predicted tau accumulation in the medial temporal lobe at follow-up, here depicted in pink. And so when we statistically compare those 2 distributions, we found that the correlations where locus coeruleus at baseline predicts changes in tau at follow-up were greater and statistically different from those in gray. So this would align with what we know from autopsy work, where locus coeruleus changes precede those of tau in the medial temporal lobe. We then wanted to know if that also had some functional implications. So we extracted these correlations from the medial temporal lobe regions and correlated them with cognitive performance at follow-up. And we saw that greater locus coeruleus related tau accumulation in the medial temporal lobe was associated with worse performance on the preclinical Alzheimer's disease cognitive composite, the PEC, both for the left and for the right side of the brain. Now, to bring it all together, we did some mediation analyses. And here we wanted to see whether there is potentially some kind of trajectory of changes, even though, of course, this is observational data and we have to be careful with any causality claims. But we tried to separate out the timelines here. So we had locus coeruleus at baseline. We had the changes in tau pathology in the medial temporal lobe, and then we had cognitive performance at follow-up. And we saw that the relationship between locus coeruleus intensity at baseline and cognitive performance at follow-up was significantly mediated by those changes in tau pathology during the measurement period. So indicating that there is a potential pathway of tau pathology from locus coeruleus to the medial temporal lobe impacting cognition. And I think that also makes sense. We know that cognitive changes are often a downstream event of cortical changes and accumulation of cortical pathology. Now, I think that's great. We now are getting closer to showing that the Braak stages can be confirmed, that we have some evidence in that direction. But it doesn't really tell us anything about how well locus coeruleus can really predict tau pathology compared to other metrics that we already have. Do we really need the locus coeruleus if we have amyloid scans, if we have hippocampal atrophy, one of the most commonly used volumetric measures of to predict cognitive decline? So we set out to compare those. And this is work from my PhD student, Nina Engels. And we decided to focus on amyloid negative individuals because, as I mentioned earlier, we are really interested in those preclinical Alzheimer's disease individuals. And so this is the distribution of all the individuals that we had available. And you can see we selected first those individuals who were amyloid negative. And then, because of course lower levels of amyloid burden can also have some noise to it, we restricted this a little bit further to make sure that we had a replication or a validation of our findings. And what we wanted to do was we wanted to compare performance of locus coeruleus intensity metrics in predicting elevated entorhinal tau in these individuals and compare it then to hippocampal volume and amyloid burden. And here you can see the performance. So these are the ROC curves. In blue, you see the ROC curves for the locus coeruleus. In red, the amyloid measures, and then in gray, the hippocampal volume. And so for both analyses, we saw that locus coeruleus intensity was the best predictor in terms of sensitivity and specificity for an elevated entorhinal tau. Then we thought, okay, that's great. What about clinical progression? Is the locus coeruleus better than these metrics in predicting clinical progression measured with the CDR sum of boxes that we also had available? And so I can tell you that for hippocampal volume and for amyloid deposition, we didn't see any significant associations in predicting clinical progression, but using the unit cutoff here for locus coeruleus metrics, we found that those individuals with lower locus coeruleus intensity had a twofold greater risk of clinical progression compared to those with higher levels of locus coeruleus intensity metrics. So again, these analyses show us that not only is locus coeruleus intensity good in its spatial patterns, in predicting the spatial patterns of cortical tau pathology, we can also predict who's likely to develop elevated entorhinal tau and progress clinically better than the established metrics that we have. And so to go back to part of the core problem statement, so far I have shown you that locus coeruleus imaging correlates with those initial burdens of tau. It precedes accumulation of initial tau according to some of our analyses, of course, bearing some of the limitations of observational data, and that it tracks with the early cognitive symptoms of Alzheimer's disease. Of course, now we want to go into trying to see if that can also give us some more insights in how tau spreads, so that if we have this marker that signals early risk, can we also use it as some kind of metric to determine when to intervene or how to intervene and halt the progression of pathology. And I'm sure many of you are aware of all the work that has been done out there looking at how tau propagation happens in the brain, and the huge focus that has happened on functional connectivity, network propagation, which of course is inspired by the fact that those stereotypical patterns of tau propagation are almost predictable, and you can see them in many individuals, assuming that some underlying brain property must guide that, and potentially this might be connectivity. Now, if you look at that for the locus coeruleus, it is important to know that the locus coeruleus fires in two ways. So it can fire in what we call a phasic way. So this is when we are confronted with new environments, when we get high aroused confrontations. For example, when somebody would drop the ball in the middle of a silent room. This is when your locus coeruleus neurons start to fire in a phasic mode. So these are high frequency bursts of activity. But then the rest of the time, the locus coeruleus neurons fire in what is called a more tonic mode. And these are these low frequent fluctuations that allow you to just go through the day and orient yourself to what you need to do. But these modes of activity have also been proven to be important for understanding Alzheimer's disease phenotypes. And this beautiful work from Omolubabi that was published 2 years ago, where she investigated mice where LC neurons were injected with tau pathology. So there were control mice, and then there were mice that were that had these injections of tau pathology in the locus coeruleus neurons. On top of that, some of those animals received tonic stimulation through optogenetics, and others received phasic stimulation. And what they noted was that those mice that had accumulations of tau pathology in, or received the tau in their locus coeruleus neurons, or also got the tonic stimulation, that those mice exhibited increased anxiety and depression. They were performing worse on memory tasks, spatial and odor discrimination tasks, and the fiber density that was emerging from the locus coeruleus to the cortex was also lower. So it seems that tonic stimulation mimicked some of the things that we typically see in the earliest stages of Alzheimer's disease. Whereas if we look at phasic stimulation, those animals did not exhibit any stress. They were behaving normally. They had no behavioral impairments, and there was no reduced fiber density in the locus coeruleus, suggesting that with phasic activity, if this is at an optimal level, that we may be able to counteract some of the negative effects of tau pathology. And this has been guiding some of our work as well. Of course, when we are doing fMRI imaging, it's really hard to go to the neuronal firing patterns. We don't have that temporal resolution. We also don't have the spatial resolution to get into those specific neurons. So what we can do is we can get some kind of proxy for phasic firing. And what we decided to do was to focus on a novelty-related task. And this is work from Kopis, who's an instructor in my lab. And so we had this task where individuals were presented with face name associations that they had seen before. So those were not new, those were repeated. But they also saw face name associations that were new. And we assumed that if you would see new stimuli, that might trigger more phasic-related locus coeruleus activity compared to the repeated face name associations, following some of the animal work that I just mentioned. So looking at these contrasts, we saw that higher locus coeruleus activity was indeed seen during these novel versus repeated conditions. We also saw higher activity in other regions that are typically associated with face name associations and memory. So we see the amygdala, the hippocampus, the fusiform gyrus, but we of course focused on the locus coeruleus here. And we noticed that those individuals who had lower locus coeruleus activity during novelty were the ones who were declining faster on memory scores. Now, going to some of the ideas of how tau could potentially propagate, we related these locus coeruleus activity levels to our tau PET maps. And we saw that lower locus coeruleus novelty-related activity was associated with greater entorhinal tau, also inferior temporal tau, and this was specific to the locus coeruleus. We didn't see this when we extracted activity from the amygdala or from the hippocampus. And again, when we put it in a mediation model, we saw that the relationship between lower locus coeruleus novelty-related activity and memory decline over time was significantly mediated by entorhinal tau pathology, suggesting that activity can be one of the mechanisms, and maybe even phasic activity can be one of the mechanisms contributing to tau propagation from the locus coeruleus to the cortex. Now, that brings me to my final point, and that is looking at pathways that confer resilience or risk. And we've been trying to tackle this in two ways, by looking at heterogeneity in the spatial patterns of the locus coeruleus, but also heterogeneity within individuals and their cognitive profiles. When I talk about heterogeneity in spatial distributions of locus coeruleus vulnerability, we are basically talking about the complexity of the locus coeruleus across its entire shape or length. And this is an older study here on the left, where you can see that loss of locus coeruleus neurons is more extensive in the rostral parts than in the caudal parts. So you can see here the difference between the younger individuals here in the dashed line compared to the older individuals is much greater here in the rostral part than in the caudal part, suggesting that loss of those neurons is greater here than in the caudal part. And then there was this beautiful study last year in Acta Neuropathologica showing that tau pathology is also more abundant in the dorsal rostral parts of the locus coeruleus compared to the caudal parts, suggesting that there may be some vulnerability within the locus coeruleus that can signal different clinical phenotypes potentially. Now, to be able to measure the locus coeruleus in such detail requires different imaging methods. And so at Maastricht University, we developed a 7 Tesla sequence that allowed us to image the locus coeruleus in much greater detail than what was possible with the 3T. So here you see our 3T sequence. It's really hard to disentangle the locus coeruleus, but when you go to our 7T sequence, you can really see the nice length of the locus coeruleus here. And so this is the method that we've been using nowadays for our 7T data. And using that data, Maxime, a postdoc in our group, has correlated plasma biomarkers of Alzheimer's disease with locus coeruleus intensity measures across the entire length of the locus coeruleus. And similar to that Acta Neuropathologica study that I just showed you, we found that lower locus coeruleus intensity was associated with greater tau in the rostral dorsal sections of the locus coeruleus. And this was true for p-tau231, p-tau181, and to a lesser extent for p-tau217. We didn't see any associations in the caudal sections. We also had total tau, NFL, and A-beta metrics. We didn't see any associations for A-beta, which is to be expected based on the neuropathology data. And the neurodegeneration metrics were a little bit more scattered. Total tau was still more rostral, but then more unilateral, while the NFL metrics were more scattered throughout the locus coeruleus. But again, confirming this spatial vulnerability that is typically associated with the Alzheimer's disease metrics. As I mentioned, we also are looking at, and this is something that we just started, we are looking at individual heterogeneity that potentially, where locus coeruleus could confer resilience. And this is inspired by some of these graphs that I just showed you earlier, where we see that individuals who have actually higher levels of amyloid are able to maintain their cognitive performance despite the levels of amyloid, but they do have a good locus coeruleus. So is the locus coeruleus some kind of metric of cognitive reserve that is protecting them against cognitive decline? And so Lucas, who is a graduate student from the Netherlands and has joined us for now a year, has been working on this. And so he created clusters of individuals based on amyloid pathology and cognitive reserve. So being education, in the MNOT scores, and professional attainment. And we saw these 3 clusters. And what was really fascinating is that the cluster 3, which is actually the blue cluster here, those individuals with high levels of pathology but high cognitive reserve decline fast if they have a poor locus coeruleus, but show strong practice effects if their locus coeruleus is intact. So it seems that the locus coeruleus may be able to confer some additional resilience in those individuals that already have high levels of cognitive reserve against Alzheimer's disease-related cognitive decline. And so this is something that we are now trying to further dig into to see if there are potentially networks behind this that are associated with this cognitive resilience. So to summarize and go back to my question, I think what I've shown you is promising. I think, yes, look-through-the imaging can become a marker that signals early Alzheimer's disease-related risk. We've shown these relationships that mimic neuropathology. They're strong relationships with cognitive decline. But of course, we need to do more. We need more longitudinal data, I think, to really understand the dynamics of that signal and its relationship to the dynamics of pathology. I'm very intrigued by understanding these mechanisms of tau propagation better, also the role of neuroinflammation. And I think there are a lot of outstanding questions that are still very fascinating to answer. And one of them is, what drives tau formation in these neurons? Why are these neurons so vulnerable? Is there amyloid, maybe oligomeric amyloid in the locus coeruleus that we cannot yet measure? And I didn't go into this because of time reasons, but we do know that there are different spatial patterns and different regional vulnerabilities that co-localize with different neurodegenerative diseases. So can that help us in going towards differential diagnostics in the future? But one of the biggest questions that I've always been asked is, what does this mean for the Jack curves? Are you claiming that this model is inaccurate? And I would say you only find a model when you have enough data to make something that is more useful. And I think that's too early. But I also would like to point out that maybe it's already in there, and maybe we do not need to fight the model. Because if you look at this area in the model, which is actually where we are looking, these very early stages that are below detection thresholds. So these preclinical individuals, we already, Cliff Jack already indicated that CSF tau is rising slowly in the earliest stages. And so maybe what we are measuring is something that is closely associated with these changes. And we may not need to really change that model. You might need to change the exact acceleration or some of the shape of these curves, but I don't think we need to change it for now until we have more data. And by that, I would like to thank you all for your attention. Everybody at the Pharmacology Brain Study, my own lab, funding. As you can see, it really takes a village to do all of this work. But yes, I'm happy to answer any questions you may have.